The second application is in pathology or surgical specimen handling. So in pathology, CIT applies mainly to excised tissue samples, for example, tumor resections and biopsies before they are placed into fixatives. CIT is very important for molecular pathology and biomarker testing, for example, ER, PR, HER2 in breast cancer. Why is CIT important for molecular pathology and biomarker testing? Number one, protein stability. For example, estrogen receptors, prostrogen receptor, human epidermal growth factor receptor 2, and Ki67. So many biomarkers used in cancer diagnostics are proteins. So if tissue remains too long without fixation, proteins begin to degrade or undergo post-translational modifications. For example, phosphorylation, dephosphorylation, or cleavage. This leads to weak, patchy, or false negative immunohistochemistry staining. For example, estrogen receptor and progesterone receptor can appear falsely negative if CIT is greater than 1 hour. Number two, nucleic acid integrity, the DNA and RNA. Molecular tests like PCR, fluorescence in situ hybridization depend on high quality DNA and RNA. So prolonged CIT allows enzymes like RNases and DNases to degrade nucleic acids, and especially RNA, which is unstable, leads to poor results for gene expression profiling, e.g. multigene assays in breast cancer. Number three, severe impact on HER2 testing. HER2 status is assessed by IHC and fluorescence in situ hybridization. Delay before fixation can cause loss of HER2 protein expression and DNA fragmentation, leading to misclassification of HER2 status. So this directly affects treatment decisions and other HER2 targeted therapies. So HER2, which stands for Human Epidermal Growth Factor Receptor 2, is a protein receptor found on the surface of some cells, and it is encoded by ERBB2 gene on chromosomes 17. So this gene carries the instructions or genetic code for making HER2 protein. In cancer, HER2 helps regulate controlled cell growth. And in some cancer, the ERBB2 gene is amplified, leading to too many copies of the gene. So this leads to HER2 protein overexpression on the cell surface. And this results into cells dividing too quickly and survive longer, hence tumor formation and progression. HER2-associated cancers include breast cancer, gastric and gastroesophageal cancers, and sometimes found in ovarian, lung, and endometrial cancers. So HER2 can be assessed using number one, immunohistochemistry, which detects HER2 protein expression on cell membranes. Number two, fluorescence in situ hybridization, which detects the ERBB2 gene amplification that confirms HER2 positivity when IHC staining is on borderline, meaning not strong enough to call it positive and not weak enough to call it negative. So because the staining or results are uncertain, the next step is to perform fluorescence in situ hybridization to check for ERBB2 gene amplification and confirm whether the tumor is truly HER2 positive. HER2 positive cancers are more aggressive with higher recurrence risk and poorer prognosis if untreated. But they are highly responsive to HER2-targeted therapies, for example, Herceptin, lapatinib, and pertuzumab. These drugs are used in treatment of breast cancers, gastric esophageal cancers. So if the cold ischemic time is prolonged, HER2 protein can be degraded, hence false negative IHC results. DNA fragmentation during prolonged CIT can lead to poor fluorescence in situ hybridization signals, leading to misclassification of HER2 status, and this can deny a patient a life-saving HER2 targeted therapy.